Welcome everyone to my channel Bio World once again. Today I've come with the C3 cycle or Kelvin cycle or biosynthetic phase of the photosynthesis. Now previous videos deals with the light phase of the photosynthesis, dealing with the light reaction, electron transport chain, process of production of ATP and NADPH. Remaining now use of those products in the C3 cycle, that is the cycle which produces ultimately glucose. Now there are a number of cycle with this C3, C4, then CAM cycle, but ultimately the main cycle is C3 cycle that takes place in higher plant for the process of photosynthesis to evolve glucose. Now the cycle was named C3 because of the first stable product or first product which is formed in this process is 3-carbon compound, that is PGA. And the cycle was named from the scientist Melvin Kelvin, who discovered this cycle by experimenting on carbon 14 isotopes of the carbon in the algae doing photosynthesis. Generally C12 isotopes is taken place, but here he took carbon 14 isotope to trace the production of glucose and to prove that carbon of the glucose is produced from the carbon dioxide. So we'll come to the cycle. The cycle is divided into 3 stages. First one is carboxylation, second one is reduction, and the third one is regeneration. Now in this first step, we need to understand that carbon dioxide is used in this process, and carbon dioxide of the atmosphere get fixed into this process. So that is why the word carbon dioxide fixation is also taken place. Like nitrogen fixation. Nitrogen is fixed into nitrogenous compound like nitrates and nitrites. Here carbon dioxide is fixed into the plant to produce the first product PGA, and the substrate is RUBP, that is ribulose bisphosphate, which is kind of a sugar or carbohydrate you can say, which is 5-carbon compound. So RUBP, with the help of the enzyme Rubisco, which is one of the most important enzyme, you have to know this. What is the full form of Rubisco? RUBP carboxylase oxygenase. That this enzyme has a very good affinity to bind with both carbon dioxide as well as oxygen. Hence the name RUBP carboxylase oxygenase. And it is the most abundant protein found on Earth. This enzyme helps in fixation of carbon dioxide with the RUBP to produce the first compound, which is 3-carbon compound called PGA or phosphoglyceric acid. Now you must be knowing that 5-carbon compound, 1 carbon from carbon dioxide, should be able to produce 6-carbon compound. But what is produced? 3-carbon compound. So where does go the other 3 carbon? The question arises here. So 2 molecules of PGA is produced in the process of carboxylation. Hence 6-carbon compound is formed by the 2 molecule of PGA. So we got the 6 carbon by 2 molecule of PGA. In this way the first process takes place called carboxylation or fixation of carbon dioxide by the help of enzyme Rubisco. So from the alcohol or from this sugar, you can say the next one is acid, and then your next one would be aldehyde production. So reduction is going on, taking place. We have come across the organic chemistry, that reduction taking place here, the process of like glyceric acid to glyceraldehyde will be coming in the next step. Now while production of PGA, the next process is evolving of 1,3-DPGA, that is 1,3-diphosphoglyceric acid, which is another 3-carbon compound. In this process you can see that phosphate is added with the phosphoglyceric acid. That means here it was 1 phosphate, but here there would be 2 phosphate, 1,3-diphosphoglyceric acid. That means from where the another phosphate group has come, that has come from the breaking down of the ATP to release inorganic phosphate. So this phosphate will be added to phosphoglyceric acid to form 1,3 diphosphoglyceric acid. That will be again 2 molecule of it, that is 3 carbon compound. So the first ATP is synthesized over here. Now as 2 molecules of DPGA is produced, so 2 molecule of ATP will be broken down into ADP and inorganic phosphate to release 2-phosphate. Next step is reduction, where 1,3-DPGA, that is diphosphoglyceric acid, will reduce to phosphoglyceraldehyde, PGAL, which is also known as triose phosphate, as same it is 3 carbon compound. So from 1,3-DPGA, what is produced? Triose phosphate, that is phosphoglyceraldehyde. That means addition of hydrogen. Addition of hydrogen means what? Reduction. So addition of hydrogen is taking place with DPGA to produce PGAL. And this hydrogen will come from where? This will come from breaking down of the NADPH to NAD plus and hydrogen. This hydrogen will be added to DPGA to form PGL, that is phosphoglyceraldehyde. So this was the second step, that is called reduction. Now from here 2 steps will go out. One, this PGL, which is reversal of glycolysis in respiration. If we go through that process, we will understand, where this PGL or phosphoglyceraldehyde will go into the formation of glucose, whereas the remaining part will go into regeneration of RUBP again, because to continue the process without any hindrance, to go on continuation of the process, RUBP regeneration is required. So this RUBP regeneration will be done again from here PGL to get converted into RUBP by the again use of energy, that is ATP dissociates into ADP, releasing inorganic phosphate to form RUBP. So in these 3 steps, the process of Calvin cycle takes place in higher plant. That is, first one is carboxylation, then reduction, and then regeneration. We need to know the intermediate product, as these are enzymatic process. The process takes place by the help of the enzyme. So this was Calvin cycle in higher plant, which ultimately leads to the production of glucose. In my next we need to understand after Calvin cycle that how many total molecules of ATP and NADPH was used in this cycle. Now we have come across that 1 molecule of carbon dioxide, that is, while fixation of 1 molecule of carbon dioxide, total how many molecules of ATP was used? Now in carboxylation and from carboxylation reduction, there was produced from PGA DPGA, that is 2 molecule of PGA will be producing 2 molecule of DPGA. While in this process, phosphate, that is inorganic phosphate, would be added to PGA. So how many phosphate would be added? 2, because there were 2 molecules of DPGA. So total how many ATP molecule was added here? Phosphate was added here. How many total ATP molecules was used here? 2. In the process of reduction, 2 molecules of ATP was used here, and one other ATP molecule was used in the process of regeneration from phosphoglyceraldehyde to RUBP. So total how many molecules we have found? 3 ATP molecule was used in 1 molecule of carbon dioxide. Now how many molecules of NADPH was used? Here from DPGA 2 molecule of phosphoglyceraldehyde was produced. So there was 2 molecule of PGL, that means 2 NADPH would be used. So 2 means from 1 carbon dioxide molecule 2 NADPH will be used. So total in 1 particular fixation of carbon dioxide 3 ATP and 2 NADPH will be used. But glucose is 6 carbon, that means total 6 carbon dioxide molecule is needed to produce 1 molecule of glucose. That means 6 molecule of carbon dioxide will need how many ATP molecule? That is 6 into 3, that is 18 ATP 1, 3. So 6 will be 18 ATP, and 1 molecule of carbon dioxide need 2 NADPH. So 6 molecule of carbon dioxide to produce 1 molecule of glucose will need how many molecule of NADPH? 6 into 2, that is 12 NADPH. So total of 18 ATP and 12 NADPH will be used in producing 1 molecule of glucose by using of 6 carbon dioxide molecule. So 6 times the Calvin cycle will go through to produce this number of molecules of ATP and NADPH. So this was the important part of the Calvin cycle, to know how many ATP and NADPH will be used in this throughout the process. So hopefully the video would be beneficial for you people. Do like, share and subscribe the channel. Thank you for watching it.